And the last presentation of this session is given by Professor Vermandel, and he will present the concept of a clinical trial where they try to treat PDT in the resection cavity after, I think, fluorescence-guided resection is the intention first? Yes.
Good afternoon. Thank you for the scientific board and Herbert to let me the opportunity to present this work. White on light blue is not visible. So my talk is about a setup of it's pretentious, the first pilot study on intraoperative 5-LI PDT for newly diagnosed GBM, so what we call the INDYGO trial. So we already know a few about glioblastoma, a few words, a few more words about it. So it counts for almost 20,000 GBMs diagnosed per year in the European community. So it's not so rare. And is the third cause of breast cancer in young adult population, and as we've seen before, the overall survival median is less than 18 months with the present standard of care. So my talk is more an outline, a roadmap to a clinical trial we are engaged in on PDT for newly diagnosed GBM. It is not on the clinical trial itself. So it's a long way to get to the first patient recruited. So we decided to describe this process. So first, we had an idea to develop a seamless approach with a standard of care to apply PDT during the standard of care. We should have to design a specific lighting device, and then to prove the efficiency of this device, to set up and to design a clinical trial. And finally, we had to undergo to the ethical committees. So just for remember, the standard of care includes surgical resection, which can be total, subtotal, which is a criteria on the MRI. So the surgical excision or resection can be done with intraoperative MRI or with fluorescence-guided resection, and then the patient undergo to radiotherapy with concomitant and adjuvant chemotherapy. So the extent of the resection, the EOR, is the key factor in the survival prognosis. We have already seen it before. And to improve the extent of resection, as I said, we can use fluorescence-guided resection. We have some talks about it this afternoon, and we can also use from a center with this kind of device, use MRI, intraoperative MRI, to control the extent of the resection. So for example, here there are some slices taken during the course of the surgery, and after the first stage of the surgery, there's still some remaining contrasted tissue that have been removed after the second MRI. Also, this MRI has the interest to update the map of the brain. I mean, the map for the neuronavigation system. So, despite the brain shift, we can registrate, we can update the map and then go again for navigation. As we've seen also this afternoon, FGR is a real progress in the surgery. As we've seen, it increased the progression. It increased the progression-free survival. We have not yet demonstrated the overall survival, but it seemed to be also interesting, since it's correlated with the extent of resection. Sometimes it may cause some neurological impairment, because we can go too further with the fluorescence, so we have to keep it in mind during the surgery. And so that's why we feel intraoperative PDT is a good way to deliver PDT since we can benefit from the FDR previously achieved during the resection, and so we can control better the extent of the resection to limit neurological issues. Intraoperative PDT might be embeddable within the standard of care and within the surgical flow. So that's why we have added this red light illumination during the course of the standard surgery. So it's just an add-on. We just switch on a red light for a while, and then we expect a PDT effect. To deliver this light, we have developed a specific light device. Here there is some kind of balloon. So this device combines an optical fiber with a light diffuser, and it aims to be suitable to the surgical cavity. For more detail, you can visit the poster 75. So the characteristic of this balloon is to be suitable to the surgical cavity, so we can inflate it with diffusing liquid, so to deliver light homogeneously inside the cavity with a specific laser device with the light bring to the balloon through an optical fiber. Obviously, to go forward to a clinical trial, we had to set up a risk management of the device according to the European Community Directive. So we have evaluated this device during the manufacturing, during teaching, where courses are given to a nurse, surgeon, and physicist. How to use it, or to storage it in the neurosurgical department, and also the end of life of the device. It's not really scientific, but it's mandatory to expect a clinical trial. Also, we have developed some standard operating procedure. We want PDT to be applied in any center, in any oncology center, not only for PDT experts. So we have to teach them how to apply this system. So we have developed a standard operating procedure for nurse, how to set up the device inside the operating room, for the physicist, how to calibrate to use the laser, and for the surgeon, how to apply this balloon in the surgical cavity. And to really ease the whole process and to avoid some consideration on time and so on, we have designed some kind of prescription abacus. According to the volume of the surgical cavity, we deliver a lighting duration, and that's it. We assume that with this kind of abacus, we will deliver 25 joules per centimeter square at 5 millimeter in brain tissues. So we really try to ease at the maximum the PDT process. From that, we have designed a pilot study with the primary endpoint. We have to start with it, is the feasibility study of intraoperative photodynamic therapy early after surgical resection for that's the novelty, of newly diagnosed glioblastoma. So the primary endpoints are no unacceptable toxicity or unexpected event. Secondary endpoints will be also measured. Obviously, the progression-free survival, the overall survival, but also the evaluation of the treatment response on MRI using perfusion diffusion MRI, and which is really important for the caregiver and the family, the evaluation of the quality of life. At this stage, 10 patients will be enrolled, recruited. In France, we have two different committees. The protocol must be approved by two different committees, which is the French Medicine Agency that will approve the medical device, and here we have a medical device and a drug. The drug is marketed for diagnosis, but not for therapy. So we have to ask for the authorization for that. So here we have the difficulty to have, in the same time, the medical device and the drug for this specific condition. And the French Medicine Agency will also validate the design of the study. In parallel, we have a specific institutional review board, more dedicated to ethical acceptance of the trial and also about information to patients and caregivers. So all these aspects are managed by different committees. So we are happy to hear last week that this part was already approved, and we should have results on the French Medicine Agency in the coming weeks. So it means that we probably be able to start this feasibility study in January, which is very interesting and encouraging. And so we will add this therapy in the course of the standard of care, which means surgical resection, intraoperative PDT, associated with the standard chemotherapy and radiation therapy. So we are already thinking of the next step. So we are designing with some partners the design of a multicentric clinical trial, which will be randomized and controlled. We are discussing with the European Medicine Agency, because it should be European at least, or in the US we hope so. And so we are on discussion on the scientific advice of this study. Always for newly diagnosed diagnostic. As newly diagnosed diagnostic as a standard of care, if we want to change the standard of care, we have to discuss with these different agencies. So we have some consortium bringing together ACMEs, academic partners, and clinical centers. For example, some ACME involved in drug management, in laser therapy, or in clinical trial design, and some center in Europe involved in neuro-oncology. Also, we have with us the European Clinical Research Infrastructure, ECRIN, which will help to manage the clinical trial in Europe. And that's it for my presentation. So please visit our posters. Thanks.
Merci bien pour ta contribution. You have questions? Recruit patients? No. If there is no question yet, I have a question to the balloon. You inflate it with different amounts of liquid to adapt for different resection volumes, or how do you handle these different expected probably volumes of resected tumor?
Yeah, so just an illustration image. So we have the balloon inflated at the beginning, so it could be inserted in the cavity, and then we can inflate it with the diffusing liquid to fit the cavity, and so we fill the balloon until there is a contact between the balloon and the surgical and the margins. You get it?
I see. Yeah, we have to talk about the homogeneity of light distribution.
Yeah, sure. Yeah.
Yeah, but there is a question I've heard.
My question was about the homogeneity of the light intensity.
So we did measurement on all the balloon surface according to different volume injected inside the balloon, and so we have something quite homogeneous. For more detail, I don't have the measure here, but for more detail, we have the poster in front of the door, and so we can discuss about it.
You made some measurements when the central diffuser is dislocated a little bit?
It could not be dislocated because it will always be that's the design of the device. There is some kind of fiber guide here, and we can see there is a transparent segment here, and the optical fiber is stopped and in this part. And this part is right in the center of the balloon. So it cannot move inside the balloon.
So foolproof.
We hope so.
Okay. Then, if there are no further questions, I thank all the contributors for their contributions.
Thanks.
Thank you again.